Hey everybody, in this video we are going to cover a brief introduction to the human genome, or at least a general overview of the human genome. So in the last video we talked about genome sequencing, and we talked about how one would sequence a genome, say if we wanted to do it today, if we had a new organism, or we wanted to sequence a human genome of a specific person, how we would do that today. With respect to the human genome, well, they used a different technique. They used Sanger sequencing, and we will talk about Sanger sequencing because Sanger sequencing is very common today for specific sequences. So let's say for determining the sequence of a small PCR product, and we will talk about PCR too. And those things, we'll talk about those in the next video. But the human genome was sequenced in about, I think, over about a dozen years or something like that. And I think that it was published in the early 2000s. And so once a genome is sequenced, so the first time it is sequenced, the first available sequence of genome sequence for a species can become the reference genome. So it's a sort of a sequence that other scientists who are studying the same species can use to compare, their sequence data to. So this is the reference genome. So everything is going to be compared to the reference genome for the species. And so there is a reference genome for the human genome. So there's a reference genome sequence. And so what does the reference genome have? So essentially it has a sequence for each chromosome, right? And there are 22 autosomes and 2 sex chromosomes. So the reference genome has a sequence. So if you went and you were to look at the reference genome, you should find at least 24 sequences. One for each autosome, X chromosome, and the Y chromosome. And together, all of these are about 3.1 billion base pairs long. So together, so 3.1 billion base pairs. It takes 3.1 billion base pairs to cover 22 autosomes, the X chromosome, and the Y chromosome. And interestingly, the reference genome, so the first complete human genome, was assembled from, let's say, genomic DNA from 13 volunteers from Buffalo, New York. Okay, so wow, how do we know that? So we weren't supposed to know this, and I forget the story of how this information was revealed. We weren't supposed to know from what part of the world the people were from. Somehow, someone figured it out, and you can now look on Wikipedia, and it seems to be that these volunteers were from Buffalo, and it's not of any one person, but it's sort of a mashup of all the chromosomes in these people to get a sequence for each autosome, the X chromosome, and the Y chromosome. I believe the same time the human reference genome was being assembled, there was a company led by a scientist called Craig Venter. And I think Craig Venter's genome was published at the same time as the reference genome assembled from these volunteers. Now, when someone or a group of scientists sequences a genome and establishes a reference genome for a species, let's say this is a chromosome, and let's say this is a P arm, this is the Q arm, and okay, let's say this sequence right here, ignoring the telomere repeats, let's say it starts with something like this. I'm just gonna make up some bases here, and let's say that's a 5' end, 3' end here. Now, someone has to decide which sequence to store as the reference. Should we store the top strand or the bottom strand? And there are some general rules that scientists are supposed to follow when they do this. But for the most part, it's the scientists who establish the reference genome. They get to decide. One strand is going to be the plus strand. One is going to be the minus strand. The plus strand is the one that is stored digitally as the reference genome. So you can infer the minus strand from this sequence right here. So if you were to go download the human genome sequence, and you could do that, and you could keep it on your computer, you should see, you know, just the plus strand for each of these chromosomes, each of these 22 autosomes and the X chromosome and the Y chromosome. And you could determine the sequence of the minus strand just by complementary base pairing rules. Of course, with 3 billion base pairs, you would need a computer to do that for you. Okay, that will become important in, I think, the next video when we talk about single nucleotide polymorphisms, this concept of the plus strand and the minus strand. So the plus strand is the sequence that's stored in the database that holds the reference genome, and you can infer the sequence of the minus strand by complementary base pairing rules. And like I said, there are some loose rules to deciding which one is plus and which one is minus, but sometimes those rules aren't always followed, and it's just whichever strand the scientist prefers, the group of scientists prefers, that one becomes the plus strand. So once you have a reference genome, you know, that's just sequence information. Like I said, you could download the human genome onto your computer, and you could sit there and you could look through the sequences, and that, you know, that might be kind of interesting, but probably not, right? Because it would be kind of meaningless. So just as important as the sequence information is for a reference genome is the genome annotation. And did I spell that right? Annotation, I guess close enough, right? Annotation. So what is that? So that is information on, say, where are the genes? You know, where does each gene in the genome start and stop? What positions, what chromosomes? Within a gene, you know, where do the exons start and stop? Where are the introns? You know, for the genome as a whole, where, for each chromosome, where are the centromeres located? Where do they start and stop? How about telomeres? You know, where do the telomeres begin on a chromosome? Or end, whichever word you want to use. Where are the euchromatic regions and heterochromatic regions? Where are all the repetitive elements, right? Where are the line elements? Where are the sign elements? Now we know very well about these, but other transposons too. Where are they located? Other things like promoters for genes, enhancers, silencers. So anything you can think about, any important regulatory element, gene, anything that we've talked about this year in this course, someone needs to go in there and determine like where those things are, where they start and where they stop, and that's really useful information to have for a genome, just as important as the sequence alone. And so scientists have been working on the annotation for the human reference genome for many, many years. I think we are on like version 38 or something with respect to the annotation and the sequence, because early versions of the genome can have errors and is continually updated with new data. And this is an example for those of you who are going to continue on with a career in genetics or any sort of molecular biology, you know, even general biology. This website is really important, will be really important to you, and this resource will be really important to you, especially if you're going into medicine and healthcare, especially if you're in a leadership role in the medical field, like you're becoming a physician or a physician assistant or an RN. So the National Center for Biotechnology Information, NCBI, is the division of the National Institutes of Health, and they have, it's just a giant database of biotechnology information. And one of the resources they have is, you know, the human genome. So you can go there and you can look up specific genes for the human genome. And in this case, I searched for a gene called BRCA1. So BRCA1, and this is a famous gene because, or notorious gene, I should say, because it's involved in, often mutated in tumors. So forms of breast cancer and other cancers often correlated with a mutation in this gene. And there's so much information on this website. So essentially what we're looking at here, this gene is on chromosome 17. And you see, you don't see any A, C, Gs, and Ts in here, right? You don't see any of that in here. We could download the sequence if we wanted to, but all of this is based on the sequence. So here's the chromosome, and we've learned about what the P arm and the Q arm. So you see these little P's right here? Okay, so this is the P arm, and we have these little numbers associated here, and these bands right here. And I think these bands, you probably see them better down here. So historically, chromosome positions were based on banding patterns, and the bands are a result of how the chromosomes bind certain dyes. And so when you look at them under the microscope, you see these bands, and those bands are historically important, and they're even carried over into our this sort of highly technical type of digital database. We're still keeping track of these because I think because some scientists must consider these important, and that's how they keep track of locations of chromosomes. I'm guessing that's why they have this here. I started studying like in the last 20 years, so this type of marks on chromosomes aren't really that important to me. But okay, so we're looking at a map of chromosome 17. And BRCA1 is somewhere in here. Okay. So we kind of zoom in here, and you can see, oh, what is this? Okay, so we have like 1, 2, 3, 4, 5, 6 different sort of diagrams here, or maps. And I'm going to guess that these are all different splice variants for different versions of the BRCA1 gene. I don't know that for sure. I'm not used to working with the human genome. I study fungi. But I'm betting these are all different splice variants that have been found associated for BRCA1. And these little hash marks right here, the little hash marks, those are exons. And in between the exons, the little lines, those are introns. And you can see this arrow right here. The arrow's pointing this way because the gene starts here and ends over here. So it goes opposite to the way the plus strand, the strand that's stored in the reference genome. And you can see we have these other genes over here, VAT1, RDN2, NBR2, so lots of other genes are here. And if you want, you could even scroll. I think there's a little arrow button somewhere where you can scroll that way. You can see all the genes that are to the right or to the left of BRCA1. So a lot of scientists have jobs where they're bioinformaticists, and they've got to annotate genomes and show where all the important genes are. And you can see how this would be really important, right? If you're a doctor studying BRCA1, well, if you just have a sequence, it's not very useful. But if you have an annotation to work with, or if there's an annotation for the genome that you're interested in, well, then you could just use a search tool. Actually, this doesn't show the search tool here, but you can enter the name of your gene, and then you could find a specific location in the genome, and take a look. How many exons are there, how many introns, and get lots of other useful information with this. So okay, so that's a genome annotation. I just wanted to give you a little bit of an introduction to what is an annotation. And once you have a sequence, what do you do with those letters? Well, someone annotates it, or you annotate it if you're a geneticist and you're interested in that type of research, and you make it available for everyone to use and to help better their research, make their research easier. So now let's talk about the human genome. I don't want to bore you with too many details, but some of the most interesting things we'll cover here. So 3.1 billion base pairs, And interestingly, most mammals are about 3 billion. The genomes of most mammals are about 3 billion base pairs. So we don't really know why most mammals have similar sized genomes. Even mouse is about 14% smaller. The mouse genome is about 14% smaller than the human genome. The human genome, chromosomes range from 45 million base pairs, so that's an M, big M there, 45 million base pairs to 279 million base pairs. There are regions of euchromatin and heterochromatin spread throughout the genome. And we're going to talk about this in more detail in a second, but only 1% of the genome is comprised of exons. 24% of genome is introns. So together, these only make up 25% of the genome. So that blows my mind. I'm going to talk more about this in a second. And only 25,000 genes. Each gene is thought to include the protein coding genes, at least 25,000 protein coding genes. So each protein coding gene is thought to have an average of 8 splice variants. We talked about alternate splicing previously, splice variants. So that means 200,000 proteins are encoded by the human genome, about. That's an average of 8 splice variants per gene, or 8 isotypes, right? Isotypes are proteins that are obtained by alternate splicing. Okay, so what do we want to take a look at next? Yes, okay. So let's take a look at the average human gene. So the average human gene is 29 kb. I could put kbb for kilo base pairs. So 29,000 base pairs long. So that's the average gene size. And that's going from the +1 transcriptional start site to the transcriptional cleavage site. Now, I'm going to put these little hash marks here. So I'm going to diagram the average gene for us. I want 9 of these things. 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, now this is the 5' UTR of the average gene. Here's the +1 site. Here is the transcriptional cleavage site. This is the 3' UTR. And probably some coding sequences in here, right? Probably some coding sequences at the end of this one and the beginning of this one. These are exons. So we have exon 1, exon 2, 3, 4, 5, 6, 7, 8, and 9. Okay, so 9 exons on average for the average human gene. Exons are 145 base pairs for an average size. The very first exon with the 5' UTR is an average of 300 base pairs long. The very last exon with the 3' UTR is 770 base pairs long. So the 3' UTR is a little bit longer on average than the 5' UTR. Most of the other exons, like I said, 145 base pairs. Introns, see how I drew them? I meant to draw them bigger. They're almost 20 times longer than the average exon. So 3,365 base pairs long. So introns are much, much longer than exons. And okay, like I said, 9 exons on average, always going to be 1 less intron, right? You can count them up if you want. 1, 2, 3, 4, 5, 6, 7, 8. So 8 introns on average. And that's your average protein coding gene in the human genome. Now let's just diagram a quick pie chart. representing the major components of the human genome. So we already talked about this. 25% of the sequences are for protein coding genes. Well, I kind of made this too big right here, but these are the exons. This slice right here refers to the introns. So this is 1%, this is 24%, and that really just blows my mind. 24% of our genome is for introns, which is, you know, they're cut out and they're so long compared to exons. And wow, why are they so long? And you know, every time our DNA is replicated in all of our cells, it's got to replicate all of this stuff right here. And you know, I guess it's probably important. It's probably important for alternate splicing, but well, it blows my mind that there's so much of our DNA are sequences that are just going to be removed when a gene's trying to be expressed, or when the cell's trying to express a gene. So also very, very interesting to me, transposons, 45% of our genome is comprised of transposons. And we talked about transposons in the last video with the line elements and the sign elements making up most of those transposons. So, wow, that blows my mind too. So 45% of every time the cell replicates, it's replicating all this. Is it junk? Wow, is all that just junk and things that can cause our genome harm with mutations? So we don't really know, you know, is there a purpose for those things? Most of these transposons are mutated or partial, and they're no longer functional. But as we saw in our last lecture, that some are functional and they can cause mutations. But most of them are non-functional. But they comprise 45% of our genome, and every time the cell replicates, every time the DNA is replicated, it's replicating all of these sequences. So we have, for other components, 3% are what are known as simple sequence repeats. So these would be those sequences that, if you remember, expand by replication slippage. Like we have a CTG right here over and over again. So any combination of simple sequence can be repeated and expand by replication slippage, and they can also decrease too. But apparently, you know, even though they can increase and decrease, for some reason, we still have 3% of our genome being comprised of these simple sequence repeats. So are these, if they're not in genes, what are they doing there, and are they important? So we don't really know if they're important, if they're forming structural roles, or maybe they're junk, maybe they're not useful. We don't know yet. So in 5% are what we're going to call large duplications. So, you know, I wish I had a specific example about one of these, but I don't have any specific example for you yet. I think in the next iteration of the course, I will focus in on and find an example of a large duplication. But conceptually, what this means is, so if this is part of a chromosome right here, say an ancestral human chromosome 1, for instance. So what the large duplication means is, okay, now there's one here it's been duplicated. Okay, so we have the same sequence here and here. Or maybe it duplicated and went to another chromosome. So that's what we mean by large duplication. Apparently 5% of our genome is comprised of these large duplications. So, and down here we still have 22%. This is intergenic DNA. So other intergenic DNA that doesn't fit into this category, that category, or that category. What is intergenic DNA? So if we take a gene, then we take another gene, and let's say that's a +1 site here, that's a +1 site here, that's a transcriptional cleavage site, this is a transcriptional cleavage site. This right here is the intergenic DNA. Intergenic. Sorry about my writing here. It's getting late in the evening and I'm kind of rushing. But hopefully by saying the words, even though I'm writing kind of messy, you can understand what I wrote. So intergenic DNA is the DNA that's found between the genes. And, you know, it's important, right? Because that's where the promoter sequences are, that's where the termination sequences are, regulatory elements. So all this stuff is important. And 22% of the genome is comprised of these sequences that are found between genes, and they're not transposable elements, and they're not large duplications, and they're not simple sequence repeats. They're unique DNA found between genes. So that's a simple pie chart of the human genome. Now, in the next video, what we're going to talk about is another really interesting thing is with respect to the human genome is that 99.9% of our genomes are identical. 99.9% of, let's say, all human genomes are identical between all of us. So if you were to compare individual A to individual B, 99.9% of their genome sequence would be identical. So on average, you know, that's like 0.1% difference between each human. That's about 5 million positions. 99% of these 5 million differences are what we call single nucleotide variants or single nucleotide polymorphisms. Most of them are single nucleotide polymorphisms. We're going to talk about these. Essentially what these means is a single base is different relative to the reference genome. So just a single base. I can phrase this another way. 5 million positions of the average human are different from the human reference genome, from the sequence in the human reference genome. And 99% of those 5 million positions are different by one nucleotide position. 1% of the 5 million are what we call indels, which is small insertions or deletions. And again, unfortunately, I don't have any specific examples of these. But hopefully in future iterations of the course, we can talk about indels in more detail. In the next video, we're going to talk about these in quite a bit of detail because these are really important for current genetic research, I should say, into human disease and things like that, and human traits. The single nucleotide polymorphisms are really important. So yeah, very, very interesting. So what makes us different from one another? You know, apparently, you know, just these 5 million positions. It doesn't seem like that many. But we're going to talk more about that in the next video. Okay, so see you in the next video.